Hello everyone, I'm Bio Joe, and this is my video on,
What are you doing?
I'm just doing my video on cellular mitosis.
You are doing a video on cellular mitosis?
Yes, I'm doing a video called Cellular Mitosis, and it's about to rock. Anyway, cellular mitosis is the process in which a cell duplicates itself. Now here, we have a cell in the stage known as interphase. During this phase, the cell carries out all its normal functions and duties. This cell is too small to duplicate just yet, so it will undergo G1 interphase, where the cell grows to normal size. Now, the cell is duplicating its DNA, as shown as gray chromatin. This is known as the S phase, or synthesis interphase. If the cell divides now, the two daughter cells will be too small, so it will become overgrown in G2 interphase. The next step in dividing is prophase. In early prophase, the chromosomes condense and become visible. The T-shape on the right side of the cell are two centrioles, which separate and a spindle starts forming. The spindle forms an aster at each pole of the cell. The nuclear membrane and nucleolus disintegrates, and the green centromere of the chromosomes attach to the orange spindle fibers. Metaphase begins once the chromosomes line up at the equator of the cell and are attached to the kinetochore fibers of the spindle. During anaphase, the centromeres of the chromosomes split apart, while homologs move to opposite poles of the cell. Telophase is where the nuclear membrane and nucleolus reform. Now, cytokinesis begins. The cell forms a cleavage furrow and pinches into two cells. In plants, a cell plate forms down the middle and divides the two new cells. So there you have it, cellular mitosis.
Well, that's nothing. Big deal.
Well, how do you clones reproduce, now that the factories are all shut down?
Well, I don't feel comfortable talking about that.
Okay, I'm gonna pretend I just didn't see that.
What's his problem?